Hi, my name's Danielle Odom and I work at the Academy of Natural Sciences of Drexel University. So I work in the watershed ecology section of the Patrick Center of Environmental Research, and we largely do macroinvertebrate sampling. A macroinvertebrate is an organism that is small, but you can still see it with the naked eye, and it's an animal without a spine. So things like mayflies, mussels, clams, all kinds of things that you typically wouldn't even think of. Today, Tanya and I are sampling in Cobb's Creek within Philadelphia. We are looking for macroinvertebrates, and we find them in rocks, and we just want to collect as many as we can, and eventually it will tell us the stream health, and it will tell us if the biodiversity is rich in this particular stream. Depending on the study that we're doing, there's so many different ways that we can sample. So we use a kick sampler, which is the net, and that is primarily used to do qualitative assessments. So it means I can pull from almost anywhere where you see riffles like here, where the water's moving fast. I can do pool areas of the stream. Whereas Tanya is using the Cerber sampler, and the Cerber sampler is used for quantitative assessments where there's an actual square area that she's pulling from, so we can tell how many, basically the density, how many macroinvertebrates can we find in this area. We have a few sorting trays and a few forceps, and we have mason jars that we hold our specimens in. We have ethanol, which helps preserve our specimens. That's more or less all that we need for our studies. Not as easy to do as the Cerber, I don't think, so this like firmer material. Thank you.
Oh,
what's that?
Did you see it?
Yeah. It's so tiny. Is that it right there? It looks like it's on the surface, but I don't recognize it as like...
Well, there it is.
Oh, yeah.
I think it's a
An
mayfly.
Really? It's so tiny.
Yeah, they're that tiny now.
Oh my gosh. It's so cute.
There it goes.
Ah!
Oh, yeah, I think it's a mayfly. Let me see if I can get closer.
I can't tell from here. It's
I thought
so
I saw
small.
3 tails. Look, there's another one there.
I think I see...
1, 2, 3.
I think I saw some kind of diptera in here, just based on the movement. Oh, is that a black fly right there?
Yes!
Everything's really tiny. So there is excitement in this pan, but you probably can't see it. So once we're done with our collections, we bring the material back to the lab, and then we do what's called a rough sort.
Hi there, my name is Tanya Dapke and I'm an entomologist at the Academy of Natural Sciences, studying streams in the watershed ecology section of the Patrick Center. After we come back from the field and we've taken our samples, which are here in this jar, we have to separate the bugs from the detritus, like the leaves and the sticks. We do that under a microscope, and then we count and we identify them. And our co-op Ben is doing that for us right now. We use these types of books, they're called taxonomic keys, to help us identify those insects. Once we know the amount of insects we have and the types of insects we have, it helps us determine the health of the stream.
Looking for macroinvertebrates is something that you can do on your own. We actually have a lot of citizen scientist programs in local watersheds where they can help scientists collect macroinvertebrates, or you could just come out on your own with your family and look under rocks. Macroinvertebrate monitoring is used as a bioassessment tool to determine the overall health and quality of a watershed. So it's really important to continually do this type of monitoring to continually assess the ever-changing river. It's a very dynamic system to track.